This is CGTN, China Global Television Network.
Hello and welcome to this Razor COVID-19 special. I'm Emma Keeling. The best and in the long term only way to defeat COVID-19 is to develop a vaccine against the virus that causes it. We've looked into the history of vaccines, how they work, how they are produced and how long we may have to wait for a vaccine to SARS-CoV-2. Severe acute respiratory syndrome, coronavirus 2 or SARS-CoV-2, is the virus responsible for the current COVID-19 outbreak, declared a pandemic by the World Health Organization on 11th of March 2020. First detected in Wuhan, China in late 2019, its genomic sequence was successfully submitted to the online resource GenBank on January 5, 2020, arming pharma and biotech companies with the data needed to race towards a new vaccine. So how does a vaccine work? At its simplest, a vaccine works by infecting an individual with a dead or weakened form of the virus or bacteria, allowing the body to use its own immune response to provide a defense. The concept is attributed to Edward Jenner in 1796. He took the pus from a milkmaid stricken with the milder disease of cowpox and used it to infect a young boy who, as a result, became immune to the related but deadlier disease of smallpox. Jenner took the Latin word for cow, vacca, and cowpox, vaccinia, to name his discovery, vaccination. Vaccines became a powerful tool in preventing certain diseases like smallpox. During the so-called Spanish flu pandemic of 1918, which killed at least 50 million people at the end of the First World War, physicians tried everything they knew to treat it, even the ancient art of bleeding. The only thing that seemed to work was infusing the blood of a recovered patient into one stricken with the disease. This showed that a vaccine may hold the answer, but identifying the virus or bacteria culprit without modern-day powerful microscopes was a near impossible task. The virus they sought was influenza, named in 15th century Italy after an epidemic supposedly influenced by the stars. There are 4 types of the virus: A, B, C, and D. The 1917-18 flu was type A, highly infectious and responsible for modern-day seasonal flu. Its structure consists of 2 surface proteins called antigens, hemagglutinin and neuraminidase. Both have multiple variants, 18 in the case of hemagglutinin, which helps the virus to attach to cells, and 8 of neuraminidase, which helps the virus to penetrate human cells. These variants are used to identify different influenza types. For example, the 1917-18 flu was influenza A, H1N1. This is the same virus, albeit a new strain that was also responsible for the so-called swine flu pandemic of 2009. But it wasn't until English scientists isolated the virus in the nose of a patient infected with influenza A in 1933, and then learnt that it could be propagated in hen's eggs in 1935, that a possible vaccine could be tested, leading to the first influenza vaccine approved in 1945. So how do we make a vaccine now? Well, we still use eggs. The virus is injected inside the egg and allowed to propagate. It's then extracted and washed with detergent, exposing the genetic material to inactivate it. The hemagglutinin and neuraminidase are purified and then syringed into your body. The vaccine does no harm, but triggers an immune response, producing antibodies that bind to the hemagglutinin. This is called the lock and key method, where the antibody is unique to the specific virus. The body now has the ability to detect that virus in the future and produce antibodies immediately. These bind to the hemagglutinin, preventing the virus from forming a bond with human cells and causing the person to get sick. So can we use the same principles to generate a coronavirus vaccine? Possibly. The coronavirus family is separate from influenza, but shares common traits. On the outside is a glycoprotein spike. This crown-like structure is where the virus gets its name. Similar to hemagglutinin in influenza, this spike protein mediates entry of coronavirus into human cells. This is the lock to which the antibody key will bind. Teams from around the world are racing to find ways of targeting the spike protein for both drug discovery and novel vaccines. This process has been dramatically sped up thanks to computer modelling based on the successful genetic sequencing of SARS-CoV-2 and its submission to the GenBank. Currently, an effective treatment is harking back to the desperate actions of 1918. Trials are showing that seriously ill patients seem to be seeing benefits following an injection of blood serum from patients who have recovered from COVID-19. Regulatory bodies all over the world are also allowing human vaccine trials far earlier than normal, but it will still take time to ensure the safe rollout of any mass vaccination campaign. The earliest we could see a vaccine is still more than a year away, maybe as long as 18 months. Therefore, while we wait for a vaccine or other cure, following the advice to isolate and observe social distancing measures is still the best defence against the virus.
That animation from Razor producer Dan Bowden. One of the shorter-term measures for containing a pandemic is testing. Iceland has undertaken a comprehensive screening program with deCODE genetics in Reykjavik sequencing the viral genome of every infected person. I asked deCODE CEO Dr. Kari Stefansson what the screening has revealed.
In spite of the fact that the virus has relatively low mutation rate, it has infected so many individuals in the world that it has had a lot of opportunity to mutate. So if you look worldwide, there is a very large number of mutations in this virus. And the mutations, because the virus stays in a community, the mutational pattern becomes specific for specific regions. For example, there is a string of mutations or a haplotype that is relatively specific for Austria, another for Italy, a third for England, a fourth for the west coast of the United States. And once you have those haplotypes, if you are sequencing all of these viruses like we do, we can find the geographic origin of the infection in every single infected individual in Iceland. And in the beginning, when the first patient identified in Iceland was identified on the 28th of February, and after that, the healthcare authorities were very focused on people coming from Italy and Austria, from the Alps, where they had been on skiing holidays. And they were very effective on containing the spread of the infection from that source. But as they were focusing on that, it is clear that a lot of people came from England carrying the mutation. And because no one was focusing on England as a high-risk area. But it is clear that the virus had much wider spread in the English community early on, much more than people had realized.
So what you're saying is there are different mutations in different parts of the world in different groups, and because of your sequencing of the virus, you're able to figure out where it originated from.
Yes, and one of the interesting aspects of this virus is how different the reaction of people to the virus is. You have individuals who say this was just like a mild cold, and then you have others who end in a hospital and on a respirator and die. And when you begin to speculate what is it that makes the vulnerability of people so different to the virus, there are two major possibilities that come up. One of them is that the sequence diversity in the virus itself may be responsible, that we may be having several strains of this virus, some of them very aggressive, other one and much milder. And we and others are, of course, focusing on trying to figure that out. And then the other possibility is that it is the genetic vulnerability, that some of us are born vulnerable, others are born resistant to the effects of this virus. And the third possibility is that there is a combination of these two. And we and others are focused on trying to figure that one out.
In terms of the testing you've been doing, I mean, we've seen very low numbers of testing here in the UK. Is it easier to test in Iceland because you have a smaller population?
There is something wrong with that statement. Yes, there are only 364,000 people in Iceland, but there are also much, much fewer people in Iceland to do the testing. Why shouldn't Great Britain be able to do massive screening with all of its resources, with a wonderful, extraordinarily good science that is being done in universities.
So what are some of the key things you've discovered about COVID-19?
First, the biggest utility of the screening has been to enable the authorities to contain the infection. By doing the screening, we are identifying individuals that no one suspected would be infected, and we have been able to put them in isolation and put the contacts of those people in quarantine.
So you've been testing asymptomatic people. It hasn't just been
We
people coming to the hospital.
have done two things. We have done testing of volunteers. We offer to test anyone who wants to come for testing who does not have clear signs and symptoms of COVID. If they have clear signs and symptoms of COVID, they are referred to the healthcare system. Then we have also taken a random sample of the population and tested. I think that what we are showing first and foremost, or we are providing an example for the world to follow, is that by using screening like this and follow it up with aggressive tracking of contact, you can contain this infection.
How do you think coronavirus will change the way the world deals with future pandemics and crises?
It is interesting, if you look at the most resourceful nations in the world, you look at the United States and you look at the Great Britain, they were completely unprepared for dealing with this. I think that people will respect more the kind of a threat that lies in a virus that mutates.
The creation of the vaccine, it still sounds like it's a long way off.
I disagree with that. It's only 3 months ago that this came up, that this virus emerged, and already there are vaccines in clinical trials in man. So I think that the companies working on the vaccine have done spectacular work. There is also a considerable hope in the development of a treatment by designing antibodies based on neutralizing antibodies in patients who have recovered. So I think that science today is extremely well prepared to take this on. And I think that science will succeed. I think we will be able to bring this under control. But we have to focus on it, and it is still a bit scary.
As countries around the world struggle to find ways out of lockdown, one of the most worrying questions is whether we can be reinfected by the virus. If so, this could cause subsequent waves of infection. I spoke to epidemiologist Dr. Eric Ding from the Harvard Chan School of Public Health.
There has been a few cases, about 100 or so cases in South Korea, in which previously infected COVID patients who have fully recovered later relapsed and had a reactivation of their virus. So it is certainly possible, but South Korea has thousands of cases and the number of relapse cases is still very limited. So I think it's a rare phenomenon and it should not be considered, I think, yet too common.
We've heard that people can catch it again in 7 days and 14 days. So is that untrue?
I think it's more of that, not that they caught it again, but that the virus was never fully cleared, because some of the early PCR tests were actually not perfect in their sensitivity, as in you could test negative, but it's a false negative. And this false negative test has been pretty widely discussed, and some people put it at 50%, as in 1 in 2. So if people previously were marked as recovered because their symptoms went away and had a negative test or 2 in some countries, it's still possible that they still harbored the virus inside of them. And later on, it reactivated when the person became a little immune weakened for some reason. So it is certainly possible, but I don't think it's a reinfection. It's probably a reactivation of latent virus.
When do you think we're actually going to be able to find out the information to say yes or no definitively on that? I mean, is it the fact that because there's so little data, isn't there?
We're accumulating more, and there's a lot of ongoing serology testing studies. And serology is testing your blood serum for antibodies. So this is very different from the direct test. The previous tests are direct tests for the virus's RNA. This is different. This is a test of your body's own immune system markers, your own antibodies that are specific for this virus, which means if you've tested positive in the serology, it means your body has built up immune markers specific for this SARS-CoV-2, the virus that causes COVID-19 virus. But the issue is, how high is high enough? Because in one recent study, 70% had high enough, but 30% had very, very low levels of antibodies, even though they previously were infected, and 5% had none whatsoever, even though they were previously infected. So in terms of whether that confers true immunity and for how long, it is unclear yet, but we will find these answers soon, within the next month or two.
In terms of anything new, have we discovered anything new about COVID-19?
I think we have. Some of the new phenomena we're trying to establish is how prevalent is it, right? Because serology will ultimately tell us how many people have previously been infected and show antibodies. But now, in immediate time, we're trying to figure out how many people are asymptomatic, whether they're pre-symptomatic before they show symptoms during their incubation time, or whether they're forever silent asymptomatic carriers. And that is kind of interesting because CDC says it's 25%, the US CDC, and hence why they recommend masks for everyone. Iceland study said it's 50%. A New York study found that all women who entered hospitals in New York who got universal testing, 80% of these women actually had no symptoms whatsoever. 80% of the COVID positive women had no symptoms. And most recently on the US Navy aircraft carrier in which they tested all 4,800 sailors, they actually found 60% of the COVID positive sailors had no symptoms either. In certain ways, that's actually could be good and bad. Bad as in there's a lot of asymptomatics who could be spreading it. So if we reopen businesses, a lot of these supposedly healthy people could be going out and spreading. And this is why social distancing is so important. But it could be actually good because it could actually mean a lot of people have been infected and have built up a large proportion of the population that may have immunity. That could be good too, because maybe herd immunity could kick in on its own. But again, this is really still too early to say. We don't even know how much immunity, given what we know about the reactivation and relapse that we sometimes see. But it could be hopeful as well. So it's good and bad, and we're learning much more.
So what you're saying, there's a higher rate of people who are asymptomatic or showing no symptoms than maybe like in SARS, for example.
Yeah, well, in SARS, it's much more unclear because we never got as much testing for SARS as we now know. The number of people infected worldwide is much higher for this virus. And SARS, by the way, we defeated without this, because SARS did not spread asymptomatically. The original SARS 1.0 did not. You only spread when you're extremely, extremely sick. This one spreads even when you don't have symptoms. So this is much more pernicious because no symptom spreading is what is the most silent and fastest spreading characteristic of this virus.
And so in terms of having a higher rate of asymptomatic people, I guess makes it difficult for decision makers and governments if they're making all their decisions based on how many COVID infections they've found.
Yeah, exactly. Because right now, in most countries and most states in the United States, they're testing not at the frontier of clinical symptoms. By frontier, I mean the moment you develop symptoms, you get tested. They're oftentimes testing much further back from the front lines of this epidemic. They're testing when people go to the hospital, or in some cases in New York City, They're only testing you if you get admitted in the hospital, not just if you enter the ER. So the fact that we don't have enough testing means that we don't have a really good sense of how many true cases out there, not to mention how many asymptomatics out there. And from the limited number of studies in which we have universal testing of an entire population, it seems it's quite a substantial proportion of the population. And so these asymptomatics are actually what seems to be potentially driving this epidemic, which again makes social distancing all the more important.
How hard is it to create this antibody test? You talked about the tests and the lack of tests, especially in places like America and the UK. I mean, what's the impact? Is there a reason that they're hard to create?
Generally, from epidemiology, we know that there's a trade-off where if you make a test very, very sensitive, which means low false negatives, the false positives go up. If you make the false positives go down, then the sensitivity declines in quality. So it's very tricky because some of the early serology antibody testing that they made in Germany, for example, they made it for a wide variety of coronaviruses, which includes the common cold. There's many coronaviruses out there. And they didn't make it specific enough for just SARS-CoV-2, the virus that causes COVID-19. And so they're trying to improve that. So I think right now we're trying to perfect this. The laboratory scientists and immunologists specifically are trying to validate these tests as optimally and quickly as possible. So we will hopefully have answers very soon in the next month or two.
There's a lot of tests out there. How do we know that they are effective?
This is why we need validated. And I think even the PCR tests for the virus itself are improving. And right now, I think on that end, we've improved that to a sufficient scale that I think the validation is very, very high and oftentimes revalidated at the CDC labs. So we're confident about that now. The issue right now is scaling up. And to scale up, it's not just the number of test kits, but also reagents. And also the new shortage in the United States, for example, is swabs, because these are not just your regular bathroom Q-tips, but Q-tips are super long that can extend back. And these lack of swab kits is actually causing a bottleneck for a lot of testing. There's lots of testing bottlenecks besides the actual test itself.
So we are still quite early on in the pandemic. There are reports of long-term fatigue, other symptoms being experienced by people who have had the virus. Some are predicting it could take as long as a year for people to recover if they've had COVID. I mean, what are your thoughts on that?
Well, we don't have a year of data to tell us if it takes a year of recovery, but it is a very long disease condition. WHO says for mild cases, it's about a 2-week of illness, assuming you have symptoms, mild symptomatic cases. But for severe, which is about 1 in 5 of the symptomatics, it is about 3 to 6 weeks. That is a very long disease illness.
Life moves pretty fast. Ideas move at the speed of sound. Technology moves at the speed of light. If you don't filter out the noise, you might miss the details. We pay attention to the details because they matter, showing you a different perspective. See the difference.
Hospitals all over the world have had to restructure to allow for the wave of COVID-19 patients. Razor producer Neil Cairns spoke to Dr. Joff Lacey of St George's Hospital in southwest London.
Before even we had the real surge of coronavirus patients coming in, there was quite a a monumental effort by everyone in my hospital to totally restructure, reorganize the hospital to accommodate what we knew would be the ensuing wave of COVID patients. And that was everything from infrastructure of the wards, converting them to intensive care units, to personnel, allowing a bit of transference of skills, cross-covering different areas to allow staff to support the intensive care units, and then really developing or amending all the procedures and processes that we normally have to care for patients. Everything had to be amended somewhat to ensure that it was fit for purpose in what was a very new world. And that whole process was dramatic and incredible in what we achieved. So we were able to develop a roadmap that allowed us not only to potentially double, triple, but quadruple the number of intensive care beds that we could provide. So, I mean, I can't underestimate what an incredible job I think my colleagues have done in developing that.
And has that restructuring worked? Is it successful, or is it still ongoing as the patients start hitting?
I think what we've all started to very much realise is the very nature of this outbreak is that it's fast moving, it's dynamic, and things change very quickly, and we have to accommodate for that. So we very much initially planned for the various social restrictions that have been put in place for, in case that didn't work, and we got a kind of almost overwhelming spike of admissions of patients with coronavirus. And as you'd have heard in the news, the social restrictions have had an effect, and there is that dampening of the rate of infection. So we've been able to, which we're all very grateful for. So we've been able to adapt for that, and thankfully, I think we've been able to cope pretty well. In terms of the actual job, I am very much on the front line, as is described regularly. So I am my role at the moment is purely dealing with coronavirus patients. I'm an anaesthetist by trade, and my department has split itself really three ways to manage the current clinical situation. So some of us have gone directly into intensive care. So there's a lot of transferable skills with anaesthetists and intensive care doctors, and many intensive care doctors are anaesthetists. And so we've been able to directly support our critical care colleagues and work in the intensive care units. A second group of us are having to continue providing urgent and emergency anaesthetic services that go on regardless. I mean, nothing stops, unfortunately, for coronavirus. So, we're needed for women in labour or delivering babies on the labour ward. They don't stop for coronavirus. And all the emergency surgery that still needs to be done needs our anaesthetic input. So that's what a group of us are doing. And then the third group, which I belong to, we're involved in an emergency response group for putting severely sick coronavirus patients onto ventilators. So this has obviously been very prominent in the news, that the need for a lot of these patients to be put onto mechanical ventilators. So that is to say, once their lungs become so badly affected that their body can't get enough oxygen to the vital organs in their body, then we see it appropriate to take over their breathing to try and improve that with a mechanical ventilator. And to allow that ventilator to do its job, we need to place a tube into the trachea or the windpipe that can allow that ventilator to be attached to it. And so that's called an intubation, to place that tube into the lungs. And that's a job that anaesthetists are particularly trained to do. So I'm part of a group of anaesthetists that are going around the hospital and attending to those particularly sick coronavirus patients, placing that tube and getting them onto the ventilators and to the place of safety, namely the intensive care units.
That's it for this special edition of Razor. We'll see you next week.